also. So we were on our journey of HBB. Hemophilus is done, Bordetella is done, and now we are left with Brucella. Brucella happens to be a zoonotic infection, which means it's an animal to human kind of a disease. And there's a Norman classification that you have, which I understand is very difficult, and you don't have to learn the entire one. So just imagine that this is all to do with animals. And we all know that when you are coming in contact with animals, you have to put on a mask. Your protection and mask needs to be on point. So what is mask? M for melitensis, A for abortus, S for sui, C for canis, ovis, nutame, mask on. Repeating, we have the famous melitensis, abortus, suis, canis, ovis, nutame, mask on. Do I have to learn the animals of all? No, some of them are common sense. Suis always means pig, canis always means dog. Now I just need to learn the most pathogenic one, and that is melitensis. Melitensis is the one that is associated with sheep and goat, and that is the most pathogenic out of these. So what exactly are the antigens? Very simple again. As I said, mask on mein M for melitensis, that has the M antigen. The next was A for abortus, that has the A antigens. All gram-negative organisms have a lipopolysaccharide, so that goes hand in hand. But something that helps in binding, you must have seen many of the books have given you a very detailed way of it. I don't want you to get into it unnecessarily confusing yourself. So for binding, these have two molecules called BVRR and BURS. BVRR and BURS are two, special chemicals that God has given it for binding, and that's it. So remember, M antigen for melitensis, A for abortus, and BVRR and BURS. Finally, what is this zoonotic disease? Zoonotic disease means it's coming from animals. Either it can come from direct contact or it can come from some milk product. So those are of course going to be the ways by which an animal can come in contact with us. Direct contact is obviously the most common. So in humans, this tends to go in the reticuloendothelial cells. Anything that will go in the reticuloendothelial cells will always cause reticuloendothelial cells is what? It's a very, very big name for something very simple. That is macrophage. It's always going to cause hepatosplenomegaly. The incubation period of this is roughly 1 month. So you can say 1 week to 1 month or 10 days to 30 days. And these patients are going to have very non-specific things. There are things like night sweats, and there is joint pain, hepatosplenomegaly. Yes, actually, if I show you a full description of what all does this patient have, he can have an entire organ involvement. So there are multiple stages. First of it is going to be a latent stage. Latent stage just means that clinically patient has latency, nothing, only the serology, and I'll tell you the serology, only the serology is coming positive. Then the person enters into acute and chronic, in which acute is the one in which all the symptoms will be seen. I just told you a triad of fever and night sweats. Then there is joint pain, right? Fever, night sweats, joint pain, and hepatosplenomegaly is there. All that we have done. But apart from that, if you do blood test, these patients also show you bleeding because there is a low platelet count. So please note, there is night sweats, there is arthritis, there is hepatosplenomegaly, plus there is a decrease in the platelet count also that you notice. Also, you must have seen that this is very dangerous just in case someone is pregnant. So they have a predilection for the placenta. Why? Guys, do you know placenta contains something known as erythritol? It contains something known as erythritol, and erythritol has a stimulant effect on Brucella. It has a stimulant effect on Brucella. So they did this study and they saw that in animals, the Brucella was growing more in placenta because placenta provides erythritol, and the same can be seen in humans also. So now you know what are the features. Now if I say head to toe, can there be meningitis? Can there be everything of the heart, that is perimyoendo, all types of carditis? Can there be all the organ involvements like hepatosplenomegaly? We know. Can all the genitals get affected? Males may epididymitis, orchitis, prostatitis. Females may of course fallopian tubes, so salpingitis. Arthritis of all the joints. Can you see the joints? They have labeled every joint over here. They've labeled the ankle joint also, the knee joint also, the sacroiliac and hip joint also. So arthritis of all these leg wala joints are there. And of course, there is involvement of the thoracic and the lumbar vertebra. There is osteomyelitis. So this is a multi-system disorder. These animals are going to make it quite deadly for us. So now you know that this is a disease in the animals and in the humans. So ma'am, you will have to tell me tests which you will do for humans and tests that you will do for animals. Of course, you're interested in humans first. So what is the media that you're going to use? I'm going to use so dekho, animals may be here, humans may be here. So I will use a media which is solid also, liquid also. Dono I will take together. So please remember, Castaneda media is a kind of a media. If I draw that culture bottle, it has a liquidy part here And this liquidy part which you have usually is the trypticase soy broth. This liquidy part is the trypticase soy broth, and then they have a solid agar over here, right? They have a solid agar plate over here. So whatever organism or sample you will get now from the patient, you will put it over here. In the liquid part, you will put the sample. After that, for example, if this was your tube, right? If this was your tube, you are just gonna invert the tube like this. This culture bottle, you're gonna invert it so that all this liquid comes in contact with this solid, and finally this is where all the growth will be seen. You've understood how you do the growth? You put all the sample in this liquid, properly mix it, and then tilt this bottle so that all this liquid comes in contact with this and the organisms will grow finally on this agar. So can I say this is biphasic? Biphasic means it's gonna have solid as well as liquid properties, and that liquid is trypticase soy broth. We don't have so much of time to do all these bottles. Now we have automated Vitec. Remember a white color dabba machine that I showed you in General Micro? That is Vitec. And this organism, because it's to do with so many animals, na? Cow, animal, cow positive. Cow beats all the tests. Catalase, oxidase and urease. This is going to be cow positive, cow for an animal. Sorted. How are you going to do that? This is a culture media. What is the serology diagnosis? There is something known as standard agglutination test. Until now, I'm only talking about things you do in human beings. I've not reached the animals. Now, guys, by now you must have analyzed, or you will do in a while, there are lots of agglutination tests that are coming. So firstly, there is something known as the standard agglutination test, cold agglutination test, microscopic agglutination test. What did I say? Cold agglutination test, standard agglutination test. I'm going to repeat this mnemonic in a lot of places, but abhi ke liye, microscopic agglutination test. And these are respectively seen in mycoplasma. Standard agglutination I am teaching you over here, that is Brucella. And I will teach you this in the next chapter, that is Leptospira. So how will I learn it? CAT, so cold agglutination test is written as CAT, standard is written as SAT, and microscopic is written as MAT. Cold agglutination, standard agglutination, microscopic agglutination, CAT sat on a mat. Cat sat on a mat, and that I've written as my Billy. Cat is Billy, na? So whenever in the exam you get cat sat on a mat, you will write my Billy. That will tell you cold agglutination for mycoplasma, standard agglutination for Brucella, and microscopic for Leptospira. The same mnemonic we had done exactly 48 hours before the NEET PG exam, and exactly after 48 hours it came in the paper. Everyone was super happy. That time microscopic agglutination test for Leptospira was asked. So cat sat on a mat, my Billy. So here I'm doing standard agglutination test. Standard agglutination test means I'm doing it for Brucella. So now let's do the details. What is standard agglutination test? Agglutination means you're going to take an antigen and an antibody. From the patient, you will get antibodies. From the patient, you will get antibody, and you will add an antigen in your lab. Of course, you will get agglutination. You don't have to learn the temperatures. Antibody from the patient, antigen in your lab. You're going to get a titer of more than 1:160. You say this is an agglutination positive. But that is positive for all antibodies. If you don't want to measure IgM, you only want to measure IgG, what will you do? You will treat it with 2-ME, mercaptoethanol. And if you treat it with 2-ME, the IgM is gonna destroy. The IgM will exit. So how I have learnt it is, okay, when you treat it with 2-ME, that is mercaptoethanol, so M for IgM, IgM का story end हो जाएगा. IgM का story exit हो जाएगा, or you can learn end ho jayega, right? So IgM will be destroyed. That is standard agglutination test that you do in humans. What is the test that you do in animals? And you can see that this is a milk, animal milk, white color milk, and I can see some of the rings, purple color rings. So what is that? That is the milk ring test. You can also do a Bengal card test and a whey agglutination test. These are all animal testings that are done. But you get a question on the milk ring test. So what do you see? You want to find out, does the animal have antibody? You want to find out the Brucella antibody in the animal milk. So in his milk, in the animal milk, you will take the milk. You are suspecting that should have antibodies. You will have to add an antigen. You are doing the same antigen-antibody agglutination. If antibodies are present, you will see an agglutination, and that is seen as a purple color ring. That is why what are you seeing? You are using the milk sample and you're trying to see the ring over here. That is why this is known as the milk ring test. So here you saw the ring, here you saw the ring. Matlab these two animals have antibodies, others do not. So that is the milk ring test. You can also do Bengal card and whey agglutination test if you want to No. What is the treatment? Now, the treatment in adults is said to be doxycycline and streptomycin IM. However, there is an alternative protocol. So, we have said that doxycycline for 45 days and streptomycin is going to be for 14 days IM. However, there is almost a 6 week hota hai, right? But WHO has also said that there is an alternative in which you are going to use doxycycline with rifampicin for 6 weeks. That is something which WHO has told you, doxycycline with rifampicin for 6 weeks. And they have said that by chance there is a person suffering from neurobrucellosis. That is another thing put up by WHO. You will use the same doxycycline and the same rifampicin and also add it with ceftriaxone. Also add up ceftriaxone in that case. So see, in most of the books you will find doxycycline and streptomycin, but it is DR, that is doxycycline and rifampicin by WHO, and additionally ceftriaxone if there is neurobrucellosis mentioned. And yes, that helps us finish HBB. We are ready for a few questions, I suppose. Quickly tell me, which of the following is identified on a milk ring test? How easy as a question. Milk ring for animal milk matlab brucellosis. What is the color? You get a purple color ring, purple blue color ring on the top. Next, in brucellosis, 2-ME agglutination is used for. You'll say, ma'am, 2-ME में mercaptoethanol. IgM का तो end हो जाएगा. So what you'll be left with will be IgG. So that was a very quick question that you had. Can you answer this for me? So tell me, all of the following are true about Bordetella pertussis except. Okay. The most common cause is Bordetella pertussis only. Fine. It may occur in children and in adults. Third, bacterial culture from nasopharyngeal swab is the definitive diagnosis. And fourth, presence of antibodies in the serum is diagnostic of the disease. So see, can it cause pertussis? Of course, that is the name. Can it occur in children and in young adults? Of course, yes. Bacterial culture from the nasopharyngeal swab is the most definitive. Is presence diagnostic? Antibodies is what is the diagnostic modality? No, I told you the Bordet Gengu medium. That was the most common. So which of the following is not very true?